Streaming all right. Let's see, I'll give people just a minute to get here. But today we get to do the fun part. Today we get to actually do the part where we're genetically modifying these cells. That's kind of what we've been working on. Let's see. YouTube started. Okay. Don't see you. Looks like everything's working. All right. So today we're going to do the actual genetic modification of the cells that we've been working on. Hopefully, everybody last week finished up their antibiotic screen and has a sense of what concentration of G418 is required to select those cells, right? So last week you should have made a plate, like one of these, selected with different concentrations of G418, which is an antibiotic, and then with the screen of those different concentrations, have a sense of how much it takes to kill those cells. Not seeing this on YouTube. Okay, let me check. Like everybody's moving. So this is live now. The connection is good. Can anybody else tell me if they can see this on YouTube? Sorry if I got screen issues. Scheduled for 11. Should start automatically. Okay, somebody said they hear me. Okay, sorry for the difficulty. Okay, alright. Maybe just give it a second. Maybe I pushed the right button. Okay. Okay, so YouTube's working now, right? Somebody say in the chat. Okay, so today we're going to be doing the actual genetic modification for the HK cells. And so this is the fun part. This is the interesting part to me. You'll have a few different things. You're still saying it hasn't started on YouTube. I think it's streaming. Check the resolution. It says I've been live for 3 minutes. Okay, it's working great. Okay. I don't know. Maybe it's. We'll figure it out later. But for right now, at least it's recorded. And it's streaming to a bunch of different places, so people should be able to do it. But okay, I'm gonna get on with it. The antibiotic screen should be done. You should have a sense. Probably it's going to be like 400 to 600, something like that. Yeah, the noise is probably the incubator. There's just not much I can do about that. It's just an incubator. But okay, so what I'll do is we'll get out some things to warm up. I'm going to do that, but you guys won't actually need to do that. So what you're going to need to warm up is your trypsin solution, your media, which will also be in a different model than that, but and then you get one of these plates because we're going to make another plate. So the plan is we're going to make another plate, and this plate will be the one that you're doing the genetic modification. And when you're setting up for that, we'll do that in a couple of days. So to start with, what you're going to do is just make a plate. We're going to let it grow for a while. So I'll go over how to make a plate again. So first thing, try to work in a clean environment. This stuff as clean as possible. The plates are worse about contamination than the flasks, because the flask is actually closed. So a few things to watch out for. I'm sure a lot of you guys got contamination. It's just, it's a skill. It takes practice, and there's a lot of stuff that you have to do to get to where you get less contamination. But there's a few easy things to remember. One, clean all the surfaces. Wear gloves. Wear masks. Clean your hands all the way up to your forearms with alcohol. Just like try to be as clean as possible. Don't take the lid off and set it down and then just leave it exposed to the air. Things like that help a lot. I'm going to grab some HEK cells. So you should have a flask like this with HEK cells. I'll show you this flask is actually a little overgrown. You see these are like 100% confluency, but that's okay. We can still use them. Let's see. So if you don't have two flasks, just be really careful, because it's the kind of thing where if something goes wrong in the next experiment, everything gets contaminated. If you don't have a backup flask, you're out of stuff. So always make a backup flask if you want to keep everything going and give yourself plenty of flexibility. And then you should have a nice backup flask that you can use to keep your cultures going, so you can never try. You should have 4 of these 12-volt plates, so you've got a few tries. Weird static. There's some weird static on the sound for this. Yeah, I don't know. I've turned everything off that was on before. YouTube is dead. This is streaming. Now a pipette so tiny. I'll answer a couple questions we're going. I'm never sure if it's all getting in the well. Should I touch the pipette tip to the media to be sure? Maybe pipette down. Yeah. I mean, if you're doing like 1 or 2 microliters, it's really tricky. You kind of get used to it though. But yeah, just definitely touch it to the media, squirt it in, pick it up, and then release. And you should be able to see the difference. This is working great. I wonder if there's not 2 streams on YouTube. Hold on. Yeah, I show 5 viewers on YouTube right now. Oh man. Then I show 7 viewers waiting. Okay, so can somebody go into the chat and tell people that there's another one? I don't know how I managed to have 2 of them. But there's one that's going and one showing upcoming, and for some reason they have the same title. I don't know. But yeah, so this is class 4. My screen went off. This came up. Yeah, it's the same microphone as last time. Hasn't changed anything. Okay. Yeah, this is part 4. Okay. Man, stream trouble. Okay. Well, hopefully people can help us sort this out. But I gotta keep going. This is gonna be recorded for the next class. Okay. So, you should have your HEK cells. You should have a backup class. What we're going to do is we're going to do a digest very similar to what we did last time. Again, between your tools. But I got to get through this first part pretty quickly. Can't imagine what would be creating that. Yeah, it's exactly the same microphone and everything. So, take your HEK cells. Pour out the media. You're going to use your trypsin. Make sure the trypsin is warmed. You want it to be around room temperature. Take 1 ml, add it to the flask, kind of slosh it around, get all the media that's still in the stuff here. Take another ml, add it to the flask, cap it, and stick it in your incubator. Sounds like bleed from another stream. Just there. Right. It could be it looks like crosstalk. So looking at this, I show people over here in the one that's live. And there's another one here where there are people talking, but I do not see anything. Like, there's not a stream here. It's just scheduled for some time what people are talking about. So, okay. If anybody knows anything about this stuff, help me out here. I wish I had some way to kill this other one. Maybe if I make it. Okay. So, YouTube works now. There's just some sort of streamless class for. Well, it is an electronic device very close to the laptop. Not really. Okay. So, okay. People say I don't know what to tell you about the audio. Yeah, I don't have an LAV mic on. It's just I'm just talking to a laptop. But okay, if need be, I'll re-record this thing. Okay, well, let's get to work. So, I don't know where I think the mic is built into the camera, so I think that was a problem. It wouldn't be working. Okay. Let's see. So while our cells are digesting, the trypsin is a digestive protein or digestive enzyme. Protein, but it's going to uh, it's going to release the cells from the bottom, and they're going to be floating around. And when that happens, we're going to transfer them to this plate. So to prepare for that, we need one of these. So we're going to fill this with media up to the 14-inch mark or whatever, not inch, milliliter mark. You won't be able to really get there with a syringe. Depending on syringes, so you can go a little past when you're filling it. And again, you're going to work with the noise might have something to do with the fact that there seem to be two streams on YouTube for some reason, because apparently I'm bad at this. But I don't want to kill one of them. I can't figure out how to do it without possibly killing the other one. And then I don't know what that would do to the recording. Okay, give me 1 second. I'll be right back. Got to grab something. Focus is also acting pretty weird today. But okay. So, with these guys. You're going to need a syringe filter. Again, try not to touch the smooth end if you can avoid it, because that's the end that's going to be going in your stuff. There is a protocol. It's in the classroom. It looks like. Oh, look like this. It'll be under class 4. Yeah, I might do it. Anybody who signed up for the class should have access to the Google Classroom, and in the Google Classroom, there's protocol for class 4. And yes, it will be up on YouTube later. But okay, before my soul don't get digested to death, make this over. All right, so you got your syringe and your filter. Take the 15 ml tube. So growth media, all 15 with FBS. You're gonna fill it up a little bit past the 10 mark. I usually don't go all the way to the very top, but leave a little bit. And then just syringe filter that into lower tube. The filter's job is to remove any bacteria or things like that that may be, in the thing. And then we're going to wait for that to finish digesting. Static just disappeared. So some people are saying the noise is super distracting. Some people are saying the static is gone. I don't know. Yeah, it might be. I don't know. Okay, audio cleared up for, I don't know. Everybody's saying it's gone now. So good job, us. I don't know what happened. Okay. Apparently it's in and out. Okay. All right. So okay, let's go forward. So make sure you keep your stuff recapped. And another thing that's important when you're handling your media and you take the cap off, try to keep the lid as the only thing that goes over it. You don't want to put the thing on and then put your hand over it, because just think of bacteria and everything just raining off of your hands and arms, especially coming out of your breath, winding up in your media, and then your media goes bad, you know. Yeah, different people have clear audio, different people have bad audios. So I don't know. Let's see. Now that we've got our filtered media, oh, now that we've got our filtered media, we're going to wait. Actually, I'm going to check it. Okay, it's ready. So you can see the cells are now sort of in the solution. You can see how it's sort of opaque and not just clear or watery looking. When you look at it under the microscope, you can see the cells are now, instead of attached to the bottom, they're now round and floating around. So this is what the trypsin does. Okay, some people are saying that when they disconnected from the video and reconnected, it cleared up. So maybe that'll help other people also. So what we're going to do is, you can see there's still some like flaky, clumpy spots. So what I'm going to do is I'm going to try and break those up. And I'm going to break those up with the pipette. So take my pipette, clean it a little bit. Okay. Put my mask back on. And take one of the larger pipettes. This is the big one. This is 1,000. I've got it set to 1,000 right now. Get the cap off. I'm just gonna pipette it up and down a bit, just a few times. Then I'll kind of spray it on the sidewall. Do that a few more times. That'll just, sort of knock down any big clumps and then break the big clumps up. And then from here, I'm going to grab all that trypsin with the cells. Oh, I'm going to put them in the 15 ml tube. All right. So what this will do is this will neutralize the trypsin. So the serum, yeah, people saying the YouTube audio is better than the Facebook audio. So maybe it's a Facebook issue. But you can see it's kind of, you can tell that there's sort of a cloudiness in there now, and that's your cells in your media. Now, so this, the trypsin's been neutralized, so it's not going to continue to digest and harm the cells because the FBS, the fetal bovine serum that's in the growth media, has factors that neutralize the trypsin. So this flask is done. Toss it. Everywhere there's a little drop or whatever, keep it clean. Okay. Now, let me get this where you can see it. It's important when you're seeding these plates. It's important when you're seeding these plates that you handle things as cleanly as possible because the plates are really subject to contamination. Yeah. So apparently the static is a Facebook thing, what it seems to be. Okay. You can take your 1 ml, 1,000 microliters, get your new tip, get your sort of cells and media, and take this plate and just sort of set it in a good space where you can work. And what I like to do is take an ml, lift the lid without really removing it, and just put it in there. Next one. And you just keep going until all of these are seated. And that will give you a nice 12-bowl plate with very little opportunity for contamination. You want to protect these wells as much as you possibly can. And then that's going to be the end of today for most people. So then I'm going to jump forward with the plate I already made, and I'll do what you're going to do in a couple of days. So what you want to do is you want to watch these plates, watch these wells, and make sure that they continue to grow and attach. And you might get some wells that are contaminated or that dry out or that generally aren't happy. And that's okay. You don't need all the wells. So the well should look like that. It's best if you put like a cup of water in whatever your like incubator setup if you have it in an enclosed space, because it'll really dry out. So check your liquid levels in the plates, and if need be, change the media. Just like suck the media out, add more media, because if the wells dry out, they'll just sort of turn yellow and all the cells will die, and that's it. If you keep, if you have like an incubator built, you can keep the, if you have an incubator built, you can keep the, you keep like a cup of water in there, and that will sort of help keep the moisture level in there high. That way, things won't dry out so fast. Use a pipette to change the media in a 12-volt plate, or just flip it over and dump it. You can flip it over and dump it, but you got to be fast. So the trouble is when you do like all the wells are empty, and then you've got to like, you know, redo it. I probably will re-record this. But so once you go through, the wells have to be refilled pretty quickly. I prefer to like drain one and then immediately refill it. Drain one and then immediately refill it. That way the cells just stay nice and moist, especially if you're not, you know, really good with the pipetting. But okay. So then this is going to go in the incubator, label it. What volume of water do you need to keep a plate from drying out in a few days? I usually take like, I don't know, a cup like that big, you know, a couple of cups of water, you know, a few hundred mLs or whatever, but it depends on how dry your air is really. So, you know, put a cup in there with plenty of water, more than will evaporate in a day or two, and then just check it every day. If it's getting low, put some more water in. But okay. And that only works in a closed environment. If you got it on a heat mat on a countertop, you're not going to moisturize the whole room. But okay, so then pop this in your incubator, you know, label it, the time, stuff like that. And what you're going to do is you're going to come back in a few days. Grab it. And you're going to look at it. Wrong side of the plastic. There we go. Okay, so this is one of the wells. And you can see it's about 70%. So what you want to do here, this is like this is about how you want them to look when it's ready. So once you go, here's another one. And so these are ready to go. These are ready for genetic modification. So, you know, I've got 4. We've already got some that are done on this, and I'll show you in a minute. So I want to show you all the final product as well. What we're going to do is we're going to start prepping for the genetic modification. Now, that's pretty easy. You're going to need your lac-Z plasmid, right, which should come in a little tube. And then it's going to be freeze-dried, so it should look empty. Maybe if you look carefully, you should be able to see like a kind of yellow string-looking thing stuck to the sidewall, and that's because it's freeze-dried, and so there's no water in it, so there's nothing to really see. You'll also get a sterile water tube, right? And this will just have water in it. This is 55 microliters of plasmid, and then you'll also have calfectin. It's going to be a really small quantity, so it's going to be very difficult to see, but it's just a drip at the bottom. And you may have, you know, in shipping, it may be like stuck to the sidewall or the top or something, so you may have to shake it down a few times to get it to the bottom. Okay, then we'll have our G418 we'll still be using, but not immediately, and you're going to have an XGAL solution. The XGAL solution and the G418 are in a couple of days after the genetic modification. So you'll seed your plate, let the cells attach, grow, proliferate, and then when they're about 70%, which depending on how many cells you start with, could be like the next day, or it could be, you know, a few days later. So the first step, I've got this plate, I've got a whole row. You only really need one row. You're going to see 12 wells, but you only really need 4. So look through the microscope, find 4 wells that are in good shape, look good. And what you're going to do is you're going to change the media. So the best way to do that is take another 15 ml tube. Take this guy, the filter. We're going to filter syringe. We're going to syringe filter some more media into that. So you make sure you're working with clean media. Now that media needs to be, you're going to use this to change the media in what's black. And that's important because you just want to make sure that the media is nice and clean. We're going to need this for a couple of things. So I'm going to do 4 plates. I'm just going to go ahead and filter me about 7 mLs. So we've got a couple other things to do. I don't want to waste a whole bunch of media, but I don't want to have to use a couple of syringes either. What am I using for disinfection? I'm just using alcohol. It's just a spray bottle filled with isopropyl alcohol. Okay, so first thing you're going to do, because of the timing, is set out. You should have a thing like this, right? A tube holder, and you should have a bag of snap cap tubes, right? You're going to set out 4 of these guys. And these 4 are what you're going to actually build your reaction. So we're going to be using plasmid DNA. And plasmid DNA, if you're not familiar with it, it's just a loop of DNA. I'm going to switch my screen sharing. So this will take 1 second, and then I'll show you the sequence of the plasmid. Okay. All right, so you should see it. All right, now the plasmid. In order to see my cursor, I got to be over here. Okay. You know, this is what you're actually putting in. This is the DNA. The DNA is, here's the sequence. Right. We'll go over this more in a second. So we're going to build the reaction first because it's got a 10-minute time. But I just wanted to show you guys what we're using. Okay, so you're going to take 50 microliters. So you're going to need to switch pipettes to the middle-sized one. 50 microliters of your filtered media. You want to use clean media because we're building a reaction that's going to go in the wells, right? So remember, this is a couple of days later. Your wells are at 70%. Everything looks good. Got a nice clean pipette, nice fresh pipette tip, 50 microliters, clean media. We're going to put 1 in each tube. So I'm going to see it a little better. Oops, all over there. But each one gets 50 microliters. And so you'll see it's got like a little drop of media, nothing too huge. Then we're going to add the DNA. So we're going to add 10 well, actually we need to resuspend it. So we're going to take 55 microliters. All right, so 55. Get a new pipette tip. We're going to take our sterile water. Pick up 55 microliters of water. Add it to the LAC-Z plasmid DNA. It says LAC-Z plasmid G418R. Add that. You can shake it or pipette it, but it should start to turn very slightly yellow. We add a yellow dye. DNA itself doesn't have a color, but we add a yellow dye to make it a little more visible, so you know when you've successfully dissolved it when we were freeze drying it. So you can shake it up, you can do whatever. Then sort of shake it down towards the end in like a whip-like motion and get everything down at the bottom. So there's our plasmid with that new pipette tip, and I'm going to go down to 10. So 10 microliters. Collect that. We'll put 10 microliters in 2 of these reactions. So 1. You really should get a new tip, but I'm going to rush because of our audio issues. 2. Okay. So 2 of them have DNA, 2 of them do not have DNA. All right. So separate them a bit. I usually just like put one on one side, one on the other. You can label them, whatever helps you keep track of it, right? Now we're gonna get the calfectin. We're gonna use 3 microliters, so we're gonna have to drop down to the even smaller pipette. 3 microliters can be hard to see. You know, I mean, practice if you need to. Get good at, you know, looking at it and telling, okay, there is something in my pipette tip. Because these pipette tips are really, really small. So 3 microliters. You can take your calfectin. We're going to add 3 microliters. Each one. So you got your 2 with plasmid DNA, and you got your 2 negative controls. Okay. There you go. Calfectin is added. Cap them. Label them. Lid back on your calfectin. This is a calcium-based transfection reagent. So the way I label positive and negative controls is usually with a plus and a minus. So plus, plus, minus, minus. Okay. Nope. Try not to drop your plasma. Okay. I'm going to set a timer for 10 minutes. Okay. Yes, I did say pipette up and down when you're reconstituting the LAC-Z with water. You know, whatever you got to do to get it mixed up. It should go from looking like a dry tube with maybe a weird yellow thing on the side looking like pale yellow water, right? Okay, so these two are now going to incubate at room temperature for 10 minutes. So this is, or I say two, these two groups, these four. Calfectin versus PEI versus lipofectamine. I mean, it's a good question. Calfectin is a calcium-based transfection reagent. It works by changing the charges on DNA, so like DNA and cells are negatively charged, and it works to neutralize those charges so that they don't repel each other and DNA can get in. PEI is similar. It's a cationic polymer, so it's like a long string that also is positively charged, so it helps to neutralize the charges, but it'll like fold into like a whole complex. Lipofectamine is a little bit different. It makes like little fat bubbles that merge with the cell membrane, and then so it's like two soap bubbles merging. It dumps whatever's on the inside of both of them together. Calfectin, we tested a bunch of different stuff. Calfectin worked really reliably, so we use that. Let's see, and it's simple. The protocols are really easy to use. Okay, so let's talk about our plasmid while our 10 minutes is counting. The plasmid is pretty simple. I'll bring it up. There's not a lot going on. I mean, it looks like a lot, but it's really not. So like you've got this section, which is the bacterial section. It's got a promoter, ampicillin resistance gene, and an origin of replication. So this section here just says, and hopefully y'all can see my mouse, but this section here just tells E. coli. It's just useful for telling E. coli to replicate it and selecting for the ones that have it. That way, you can make more copies of this plasmid easily using E. coli. This gene, so this is our SV40 promoter, and you see neomycin, kanamycin resistance. This is actually the G418 resistance gene. So it's the same gene that makes resistance to kanamycin, neomycin, and G418. And this is the terminator. So this gene is what allows the cells that have been modified to survive in the high G418 environment. So we last week killed off all the cells with G418, find out how much we need to kill them, and this will allow us to select for those that have been modified. So the cells that take up the plasmid will have resistance to G418 because of this gene. It's an enzyme that breaks down G418, and those cells will survive while the unmodified cells will die because this process is imperfect. So it's going to modify some of the cells in each well, but not all of them. And so that means, you're going to end up with cells that are modified and cells that are not modified living together, and you want to sort of bias it towards the cells that are modified. So you want those cells to like live and be happy and reproduce a lot and make, more modified cells than unmodified cells. That way, when you do the last part, it's a strong signal. So this part we can ignore for right now, but this is really the interesting parts. This is the promoter gene and the terminator signal. But this gene is LAC-Z, so that's beta galactosidase. It's a bacterial gene. Here's the sequence for it. You can see the protein and DNA sequence. But what it does is it breaks down a sugar called galactose. Oh, shoot. I've not been sharing the screen. Well, sorry, guys. All right, let me hit you real quick again. Definitely got to re-record this. This has turned out terrible. Okay, so here's that E. coli portion, right? Ampicillin resistance, ampicillin resistance promoter, origin of replication. Here's the G418 resistance gene and its promoter, terminator. And here is LAC-Z, which is an E. coli gene. Promoter from cytomegalovirus, the gene itself, and then the terminator. So this gene, LAC-Z, right? There's the LAC-Z DNA sequence. Big gene, kind of. Okay, so beta-galactosidase breaks down a sugar called galactose, and what we're using for the last step is something called xgal, which is like a modified galactose. So what happens is normally galactose is involved in digesting lactose because galactose is part of lactose. So like take like a galactose and a glucose and you stick them together, you make a lactose, which is a milk sugar. So you break that down with lactase, and then you have beta-galactosidase, which breaks down the galactose. But none of that really matters for what we're doing because we're not actually trying to digest the sugar. I mean, we kind of are. But what happens is HEK cells can't break down xgal, which is this stuff. It's a chemically modified sugar that basically has like a blue substance bound to the sugar. And when you break down the sugar, the blue substance is freed up to become blue. And so the normal HEK cells, if you add xgal to them, they don't do anything. They just sit there with xgal floating around and nothing happens. And the ones that do have the LAC-Z gene have this extra enzyme, and it starts to break down that xgal, and as a result, it turns things blue. And so that's how you can see which cells have been modified and which cells have not been modified. And let's see, how are we on our timer? Okay, about 3 minutes. Okay, so what we need to do is change the media in the wells that we're going to modify. You don't have to change the media in all of them, but do change the media in the ones that you're going to be adding these 4 to. So you go through, you pick the 4 you like, and you got extra wells, so pick the healthiest and like best ones. That's going to give you the best results. And for me, since I've already done this twice on this particular plate, I'm going to take the middle ones because, okay. Run out of paper towel. Okay, now we're going to use the media that we filtered. And I'm going to remove the media. And replace it. And just have something nearby that you can dump the excess liquid in. But this will just make sure that everything's happening from a really clean place. And you probably want to change tips between all of this. Make sure you don't have any sneaky contamination that winds up in places you don't want it to be. But okay, so we've changed media in all of those wells. That's the first step to the transformation. So first step to transfecting these cells, change the media in the wells. Now those wells that you change the media in, so it's a good question. I'll get to it in 1 second. The ones that you change the media in, those are the ones that have, those are the ones that you're going to be using. So here's a question. Wondering how the pipette tips stay sterile. It's just because they're down there underneath, and there's not really like anything going on down there. You don't touch them. Try to keep the lid closed to your box as much as you can. But anything that's going to get contaminated is probably going to be the top, which is not the part that touches anything. And you shouldn't get liquid all the way up to the top near where your pipette's at. But no, there's nothing about them that keeps them sterile. If you touch them to something that's contaminated, they'll be contaminated. Okay, so those are changed. I got 10 seconds left on my timer. Let me go ahead and stop it. Okay, so now these are ready to go. What you're going to do is you're going to mark. Okay, so you're going to mark which plastic you're going to use. So just put your plus, minus, minus. Okay, and that's going to give you, which ones you're going to use the plus for, which ones you're going to use the minus for. Remember, pluses has the DNA, negatives did not have the DNA. We're going to take, so we used about 70 or so. Yeah, it's like 50 plus 10 plus 3, so, make sure you got plenty of room to pick up the whole reaction. Like that's it. And then you're just going to add this to the well. So, again, you're picking up the ones that you've changed. Pick up your plus, add it to a plus. Doesn't really matter which one. New tip. Take your minuses. These are negative controls. So what you'll have is at the end, you'll have negative controls, which you can also add lac Z to, or you can also add okay, that's our 4. We've added 4 reactions, 2 with DNA, 2 without DNA. That it comes from the wiring. I mean, it could be. I really don't know. I could like gotta move this. I don't know if that helps. But okay. Let's see. So that's it. So that's our reaction. You're going to take this. You added the calpectin and the DNA, and you're going to put that back in the incubator. You're going to leave it for 24 hours, and you're going to change media again. So pretty much any transfection reagent is going to have a certain amount of toxicity. So the calpectin and all of that itself is going to be harmful to cells. So anytime you put these kind of things, you're not adding any G418, you're not adding anything like that yet. But just the transfection reagents will be toxic to the cells. That's okay. You're going to take that and stick it in the incubator. This will give the cells time to take up the DNA. And then, but tomorrow you want to change the media because you want to get rid of it. And you're just going to use regular media. Then you're going to wait 2 days. That 2 days is going to be time for the cells to recover. You'll probably notice that your cells look a little stressed. So after the transformation, they're probably going to look a little more rounded and like they're not proliferating well because this is hard. So you'll change the media in all 4, both the ones with the DNA and the ones without the DNA, and just pull it out, put a new, fresh piece of media, and then your cells will now have their new genes. Then you're going to give them 2 days. The reason you're giving them 2 days is a couple of things. One, you're giving them a little bit of time to recover, and you're giving them time to express the gene, right? So they got to like read the DNA and then make the enzyme and then make a good fit. Then we're going to move on to the last thing. Now, you're going to take your G418, which is, so remember this is today, give you a timeline. Today, you set a new plate. Wait a couple of days until it's, right. Until the cells are about 70% controlled. Then you're going to add the calpectin and DNA. Then you're going to wait. So you're going to change the media the next day, 24 hours later. Then you're going to wait a couple of days and let them recover about 48 hours. Then you're going to do this one. So you can take your G418 and you'll have discovered it's probably 400, 600, somewhere in there, whatever that range is. And then you're going to have your XCal solution. And so whatever range you discovered is the issue, or is like the good range, enough to kill all the cells within 3 to 5 days. That's the concentration that you're going to use as your selection. And then for the XGAL solution, we're going to use anywhere from like 10 to 20, but 15 should be good. The more you use, the darker it is, but you don't have a whole lot to start with. So, what you can do is you can add both G418 in whatever concentration you discovered, and about 15 microliters of the XGAL solution. And then just after you add them, you just kind of give them a little stir, and then pop it back in the incubator. The next day, you added your XGAL to every well, but what you should see is the ones that have been modified will have turned blue, and the ones that have not been modified will not have turned blue. Because, you can see it here, try to get it on an angle where you can see it. Let's try this. Oh. Does that look okay? Yeah. So you can see, like, these two are blue, so these are the ones that I genetically modified. So the media has turned blue. These two, I also added the XGAL to, but because they don't have the LAC-Z gene, they can't break it down, and it doesn't turn blue. Same thing with these. I added 10 microliters to this, so it's kind of a purpley thing. But the purple can happen, it's just because it's like the pink plus the blue, right? But the purple can happen if you use like a little bit of XGAL, or you don't get a really efficient transformation, or if you don't have a whole lot of cells, right? And so it's like really blue. Yeah, you should be able to see that. Yeah. And it'll be like really brightly blue. And it's hard to get an angle on this. I've got some pictures we'll be putting in the classroom, but it should be dramatic. And honestly, if you did it right and you wait 48 hours, you add the XGAL, and it just becomes like it starts to turn blue within like an hour usually. So you can watch it and come back and check, and come back and check, and you can watch it get blue. But yeah, I'll stick this under here, and I'll show you. But yes, so this is a good question. The cells themselves do become blue. Just want to share the screen right now for some reason. Okay. Now these are ones I did a few days ago, so it might be mostly dead cells. Yeah, see how stressed out they are? This is just a way overgrown culture. As you can see, they are blue. Let me zoom in a bit. Here, see? So the cells themselves are blue, even though they're obviously super stressed. This is too old. Maybe switch to this one. A little more. Have some more lively cells. Yeah. A little less blue. You get bluer over time. But also when you're super zoomed in, it's hard to see. Just focus. Yeah, hard to see, but you can see they're blue. You can compare that to controls, right? You can see the transformation itself is hard on them, but that's the negative control for that one. Got the same amount of XGAL solution and everything. They're just growing better. But they're not blue. And this one, blue, but it's also pretty damaging. But it shows you, even if the cells aren't happy about it, they've been genetically modified, and that they've expressed their new gene, which comes from a bacteria, and you're putting it in a human cell, and they make the new gene, and they can now digest something that they couldn't digest, so they break down the XGAL, and it turns blue. So remember your timeline. It's going to be today. Yeah, I'll probably end up re-recording this whole thing because the audio is bad, but we'll figure it out. Yes, so it's also YouTube chat. I'm using Restream, so all the chats come together here, so I should see it. Except for that other YouTube stream that for some reason is weird. So today you're gonna make a plate. So digest out one of your flasks. Make sure you have a backup. Take that, put it in your 12-volt plate. Stick it in the incubator. Give it a day or two to grow. Keep checking it. Make sure that you have about 70%. It's okay to go a little over that. I wouldn't go under that though. Then once it's there, you're gonna change the media. You're gonna add your DNA calpectin media solution that we made. Remember to let it incubate and go through that process so that the calcium and the DNA have a chance to work together. And then once you take that, add it to your wells, then change the media the next day, and then give them a couple of days to recover from the transformation, and then add the G418 and the XGAL solution, and then give it another day for everything to be fully cooked. But that way you can, it should be pretty dramatic. Let's see, after modification, will they be stressed or will they recover eventually? So they seem to be, yeah, they recover. They recover. So the XGAL just added doesn't seem to be toxic, but it seems like the breakdown products are a little bit to them. They don't seem to like being this blue. But if you don't add that, they'll just retain their thing. You can actually continue to select them. If you select hard enough, you can create a stable cell line, and the stable cell lines are created. So basically, you just modify it, and then you just keep selecting, and you can keep this cell with this modification for generations and generations. But then when you add the XGAL, it breaks it down, it's probably toxic again. But if you change the media and give them some time, they'll recover from that also. So, you know, it all just kind of comes down to like how you treat them. They don't like being in the blue state, really. I think but it's also a ton. Like normally when you're doing HK cells, they fix the cells, so they don't do it while they're alive. And even when you're doing bacteria and making them turn blue from G418, it's just way less. So to make these guys turn this blue, we're actually using a ton of XGAL. You know, it's actually a really, really large amount, and so they just don't like it. Let's see. I wonder if you could use this. Maybe. Or if you could use this blue-white screening for measurement of gene expression used for microscope for some program to measure the I mean, possibly. The tricky part is because it's an enzyme, it's also really time sensitive. So the same amount of modified cells will give you a different result an hour later, right? So because expression of this plasmid is going to like peak at a certain time, it's really time sensitive, and it's sort of exaggerated by the fact that it's an enzyme, which is why we didn't use like GFP or something, because this is going to be a stronger signal. Because you can sit there with half the enzyme and wait longer and get a stronger signal. So you have to really build in the timing just perfect, and the number of cells and all sorts of stuff. You probably could do it, but it'd be tricky. The modified cells changed. Let's see. With the modified cells change media color for the blue after a certain amount of time and growth. Start to turn purple and slowly start to edge towards a different color. So when you add the XGAL, it won't immediately change. It will just be pink. You stick it in the incubator and you'll come back and it'll be like, you'll look at it. If you come back in like 30 minutes, you'll be like, it might be slightly less pink than it was. When you come back in a little while, it's like vaguely purple. It gets purpler and purpler and purpler and bluer and bluer and bluer until it's like a really rich, sort of gold blue. Um, but it also depends on how much you use. So if you use more Xgal, you get a faster, um, darker blue, right? So it's so the Xgal is the blue stuff, right? So so they won't turn blue without it, and then once it's um, broken down, it'll like peak, and that's as blue as they're going to get unless you add more Xgal. But then when you change the media next time, if you don't add Xgal, it won't be blue. So the cells might take a little bit longer to get less blue because they're like stains now. But yeah, I mean, they won't just continuously do it unless you continuously add Xgal. And yeah, let's keep the questions to the class subject here. That's what we're here for. If you have other questions, hit me up on Facebook. But okay, any other questions while we're going? So this is going to be a big week, so make sure you're ready for it. Because if you miss your timing, like you do the genetic modification and then you wait 5 days, then you add the Xgal and then over. If you don't have time to check on your class or you're on your plate, make sure that your wells are alive and you got time to do the things each day that you need to do it. And again, the protocol is in the classroom and it lays out what the days are, so you see the plate, keep track of it. When they hit 70% confluency, that's about 3 to 5 days. Replace the media, go through the thing, the amount of like DNA, calfectin to add, and then let them grow overnight. Change the media, add the DNA, add the stuff, but don't change the media. Okay, yeah, yeah. Next day, change the media, and then hold on. Okay, so the media change color after the genetic transformation. After a while, then would it change much? Like how generally change things over the pH? I don't know. I mean, if you just let it sit there, like after it turns blue, so the pinkness and yellowness is a pH sensitive dye. But honestly, the blue from the Xgal seems to be like an even stronger dye. We're using a ton of it. So I don't know if it would ever turn green, like if the pH got low enough. Um, like, and then the pH sensitive dye goes from, you know, trying to be pink to trying to be yellow, and then it's like yellow plus blue except pink plus blue. Maybe one of the purple ones would. Um, but yeah, it's just hard. It is harder to keep track of like when you should change the media. But honestly, by the time you see blue, you're also done with the clash, right? So like, that's the output signal that you were successful. You should see like a really bright blue, and your control wells will still be pink. If your control wells turn blue, you probably mixed your tubes up. If all your wells stay pink, you mess something up, and you got some kind of an error. If you're um, you know, so just make sure that you don't add DNA to your controls and like that stuff. But it should be pretty obvious. Like that's why I wanted to make this change to the kit, because I wanted to make it really really obvious with a really strong signal. So each genetic modification is time sensitive to each type of modification. So yes, um, that's a complicated question because it depends on the cell type you're using, because different cells have different sort of speeds that they live at. But it also depends on the promoter that you're using. So remember, we're using the cytomegalovirus promoter, and it's a strong promoter, but it tends to make expression that's really strong but short-lived. And so you get like a really short spike that's like day one, day two, day three, and so it starts to produce a lot of it. But because this is an enzyme, it's just gonna, and it's a pretty stable protein. That's another thing, protein stability. it'll just float around in the media, right? So, some of it's in the cells, of course, but a lot of it's also just in the media from cells that have leaked or whatever. And so as soon as you add it, it just immediately starts changing. Most of it's in the cells, but once you go through that, um, because it's an enzyme, you know, it doesn't get used up, right? So uh, enzymes act like catalysts, right? So the enzyme like finds a X-gal, breaks it down, then finds another one and breaks it down, finds another one and breaks it down. So if there's like billions of these enzymes, they can just churn through lots and lots and lots of the X-gal over time really quickly. Um, let's see. Where some other protein might only be like a one-for-one expression. How long will the plasmid stay in the cells and the culture? Is it integrated into the cell's DNA itself, passed down through cell culture? It's a good question. So, generally speaking, circular DNA doesn't integrate, but nothing is 100%, right? So if you do enough cells, some of them will randomly integrate. You can design it so that it's more likely to integrate. You can use things like CRISPR, and you can use linear DNA and homology arms and all this stuff to try and make it more likely to integrate. But for the most part, it will just stick around as a plasmid. But um, a couple of different things can happen. So cells can silence plasmids because they realize this is not uh, human DNA, and just shut the plasmid down. So that's a thing that can happen. So even if they have it, they can stop expressing it. What we're doing mostly is a transient transfection. So that means it's going to be a short-term expression. You can just keep selecting really hard and aggressively. And what happens is, as the cells divide, yes, it's diluted, but you'll still have some cells that have the G418 resistance. So those cells will survive, and those without it will die. And so you'll end up just constantly selecting for it, but it will make it much harder for your cells because they're not like bacteria that can't just replicate the DNA. Now, if the DNA does integrate, like if you keep selecting hard for a long time and randomly the plasmid integrates, then it can become stable and it'll actually integrate into the genome just by chance and stay there, and then you can just keep reproducing those cells, and they'll just keep reproducing that plasmid because now it's part of their genome. But generally, this is a transient transfection. So, like if you just stopped using G418 and just grew them, put them in trypsinize them out of the well, put them in a flask, kept growing them, it would be gone after a while, and it would just you would add more X-gal later on, and nothing would happen. So we have a lyophilizer here. It's like a vacuum with a chiller thing and a whole setup. But yeah, so we just pipe that plasmid into the thing, add the dye, stick it in the machine, and freeze dry it. So yes, this plasmid does have an E. coli origin of replication. Show it to you. And the plasmid sequence should be in the classroom. Let me make sure this is popped up over here. Okay. I want to repeat it last time. Map. Okay. So that is our E. coli origin of replication. So that means we can take this plasmid and grow it in E. coli, and then make more of it if you wanted to make more of it. So if it didn't get silenced, wouldn't the plasmid pass from one of the cells to it? So it's not just one plasmid. So lots and lots of plasmids are going to end up inside any given cell. So you'll have, whatever your cell is, you'll have maybe 300 plasmids in it. It'll be kind of random. And then when it divides, maybe you get like 150 in each, and so on. But yeah, eventually you will get down to where there's like maybe one left if you continue to expand the culture. But yeah, there's more than one, so it doesn't usually present a problem with at least the first little while, like first 2 days of replication. It's not a problem. Okay, so that's another good question. No. If you put this in E. coli, it will not turn them blue. I'll show you why. So, this is a bacterial promoter, the ampicillin promoter, and it promotes this gene, the ampicillin resistance gene. That's beta lactamase, and that breaks down ampicillin. And so you can take this plasmid, transform it into bacteria, also using calcium. I don't know about calcite, but using calcium chloride. And then transform it into E. coli, select for them with ampicillin, and you'll get bacteria with this plasmid. But this gene, or this gene here, LAC-Z, which is the beta galactosidase gene that breaks down the X-gal and turns things blue, its expression is driven by the CMV promoter. So this promoter is a mammal virus promoter. So this is actually from a human virus, and so it's designed to tell human cells to make whatever gene is downstream, right? And just everything else about this is coded in such a way that it will express in human cells. Now, actually, we do have a T7 promoter, so it's probably not impossible to get it to express in the right E. coli. Because T7, I think it would express in BL21, so you might be able to get it to do that, but there's not a terminator, so I don't know how it would do. So there might be some expression if you got it into the right one, but this is mostly just in the sequencing side. But I mean, yeah, so you should be able to, but if it's just got this promoter, then it won't express any coli. It won't express it. But yeah, with this promoter here also, in the right strain of E. coli, you could probably get some expression, but it won't be great because they're not a terminator, unless there's a hidden one in there somewhere. Oh geez, I'm not sure what's going on. Okay, let's do this. All right. So, all right, this promoter, CMV promoter, that's a human cytomegalovirus promoter, and it's from a human virus, and so it will drive expression of what's downstream of it, which in our case is the LAC-Z gene. That's what makes the enzyme. But there is a sneaky little promoter in here also, T7 promoter, that will express in E. coli that have the T7 polymerase in it, which is DH5 alpha won't do that, but BL21 will. I don't know if BL21 requires an inducer for T7, but yeah, any bacteria that will drive T7 should make this. Whether it's coded right for it, like the codon's fine, but like there's not like there may not be the right binding sites, and there's not a terminator that I see. So I don't know. I mean, you might get some really weak expression in E. coli because of that terminator, if it's the right E. coli. Yeah. Somebody's saying stuff. Let's see. There's T7. Could do it in BL21. Yeah. Okay, so I can check on that. Everything should be on its way. Sometimes stuff gets stuck in the mail, especially with COVID and whatnot. You get a lot of disruptions in shipping stuff because everybody's shipping stuff. But if it takes too long, we can try again and try to ship it in a different way. But yeah, as far as trying to express the LAC-Z gene in E. coli, there's better ones. Right? There's much better plasmids for trying to do that because they're really just good for it. But okay, any last questions we've been on here? little longer than I wanted to. Sorry about the audio issues. I'll try to figure some of this stuff out. I'll see what the recording sounds like. Maybe the recording's better than the live. If not, I'll figure something out afterwards. Any last questions? Alrighty. So why are the other promoters in the class? Okay, so if you mean like this promoter is here to promote ampicillin resistance, so that you can replicate the plasmid in E. coli, just for the sake of manufacturing the plasmid. This promoter is here to make. This is also a simian virus 40 SV40. So that's a mammalian promoter, animal promoter, and so this will drive the expression of this gene, which offers G418 resistance, and then this one is the promoter for this gene in our human cells, and then this one is mostly here as a sequencing site, but it's also a promoter. So, like in theory, you could do some expression probably. I've never tried it in BL21. I could try it, but I don't see a terminator that work for us. Which that doesn't mean you won't get any expression; it'd just be weird. But yeah, so this is just here because the plasmid. So this is built on PC DNA 3.1 plus, right? Which is like a standard expression plasmid. So you know, when you get these kind of plasmids, there's like a cloning site, and you just cut and paste in the thing, and so they have different options on these things. And so the only thing we really had to add was black seed. Let's see. Yeah, I thought you might need IPTG induction with BL21. Yeah, but I mean, you can always add IPTG. I mean, that's not really a big problem. Just add it to the plate. But even then, without a good terminator, I don't know. Okay, sorry about the static. Looks like that's the last questions. You know, one day we'll be able to do these classes without without having to stream stuff, so then we won't have so many weird issues. But I look forward to seeing everybody's pictures. I want to see like some nice blue. Yeah, bad it is. It's a really nice blue, blue wells. But yeah. So, alrighty. Yeah. Okay. So everybody have a good day, and I'll see if I can figure out how to fix this thing. Maybe I can edit the audio or something. We'll see. All right. Everybody have a good night.